Hello everybody, we'll talk about the sensory systems. First we divide the sensory systems into two types. Okay, so sensory systems are divided into two types of sensory systems. One is called somatosensory system and another is spatial sensory system. Okay, somatosensory system includes touch, pain, temperature, those sensations you feel throughout the body. So somatosensations somatosensations include touch, pain, temperature, and those are processed in the system that is called somatosensory system. As I told you, you have these sensations throughout the body, so widely distributed. So in case of somatosensory system, the sensations are widely distributed. Okay, for example, in your skin you have touch receptors, pain receptors, temperature receptors everywhere in the body. Okay, so the receptors are present throughout the body in case of somatosensations. Special sensory systems process special senses. Special senses. Special senses include your sight or vision, your hearing or audition, your smell or olfaction, your taste or gustation. So now you see one difference between somatosensations and special senses. In case of somatosensations, the receptors or the sensations are present everywhere in the body. In case of special senses, you have highly special organs where the receptors are localized. For example, for your sight, you have the eyes. For your hearing, you have the ears. For smell, you have the nose. For taste, you have the tongue, right? So the receptors for sight are located in the retina of the eye, only inside the eye. For hearing, the receptors are only located inside the ear. For smell, inside the nose. For taste, in the tongue or inside the tongue. So highly localized, not widely distributed. Okay, highly localized. So we will talk about the special senses, sight or vision. So the sight or vision is processed in the visual system. So we'll talk about the visual system where your vision or sight is processed. First, we'll see the parts of the visual system. The visual system has a central part inside the central nervous system and central part is inside the central nervous system, and a part peripheral part are your eyes, two eyes. Okay. So we'll talk about the eye and its parts. We'll talk about the chambers inside the eye or eyeball, layers in the wall of the eyeball. We'll talk about the muscles that move the eyeballs. Those are called extraocular muscles. We'll talk about the regulation of light that enters into the eye, how that amount of light is controlled. Then we'll talk about the most important structure inside your eyeball, that is the retina of the eye. Why this is the most important structure? Because the receptors are located in the retina, and these receptors are called the photoreceptors. Okay, photoreceptors. Probably you have heard photoreceptors are rods and cones. Two types of photoreceptors, both are present in the retina. Okay, so without those photoreceptors you won't be able to see. So that's why retina is the most important structure inside the eyeball. Then we'll see how the signal from the retina is taken to the brain. That's the visual pathway from the retina of the eye to the brain. So the eyes are the peripheral organs for your visual system, and each eye has a number of things. Eyeball, which is the most important and the largest part. Then you have other structures: eyebrows, eyelids, conjunctiva, lacrimal apparatus, and extrinsic eye muscles to move the eyeball. So this is the most important part, right? Eyeball. And we'll see those structures here. If you see the eye, this is the eye from the front. You can see those structures here. Only a small part of the eyeball you can see from outside. Most part of the eyeball is located inside the orbital fossa. So you see here, you can see the sclera, this white part. This is called the sclera. It doesn't allow any light pass through it. It is opaque, and most part of the eyeball is white. Then you have transparent part. This is called the cornea. And you see the dark aperture, that is the pupil. It is actually a hole. So this is the hole or aperture or opening, that is the pupil of the eye. It gets bigger and smaller. Constriction and dilation or dilatation of the pupil occurs. Okay, and you see this under the transparent cornea, this colorful structure, which is actually a muscular structure, this is called iris. Okay, So under the transparent cornea, you have the iris, and in the center of the iris, you have that aperture or opening, that is the pupil. So pupil is an aperture in the center of the iris. Okay. So those are few things you can see from outside. The color of your eye is given by the iris. So you have the pigments in the iris, in this muscle, and different people have different colored pigments or amount of pigments, so that determines the color of the eye. If someone has blue pigments more, the eye is blue, brown or black or even green. Extrinsic eye muscles are the muscles come from the bones uh, around the eyeball, and those muscles get attached to the eyeball. So this is the eyeball, and this is the cornea. Okay, so this is the cornea, the front part of the eye, the transparent. This is a sclera. Anyway, so a number of muscles come from outside to move the eyeball uh, in different directions. So these are the extrinsic eye muscles. So they come from the bones around and get attached to the eyeball from outside. So by the contraction and relaxation of these muscles, your eyeball moves, and these are the extrinsic eye muscles. Now there are 6 extrinsic eye muscles. Uh, 4 are rectus, that means those are straight muscles, and 2 are oblique muscles. Okay, oblique muscles go like this way. So this is one called superior oblique, and there is another under, that is inferior oblique. And now you see that oblique muscle, since it is going this way, it rotates the eyeball, and these are rectus muscles. They move the eyeball up or down. By the contraction of rectus, you move up, down. There is another in the side, so lateral and medial. Anyway, so these extrinsic eye muscles, there are 6. 4 rectus, uh, and rectus help to move the eyeball up, down, lateral, medial, and oblique muscles help to rotate the eye. So here you see, this is the eyeball, and this is one straight muscle above the eyeball, that is superior. Superior means above, rectus. And there is another one below the eyeball, that is the inferior rectus. This is lateral to the eyeball, so that is lateral, straight lateral rectus. And there is another one on the medial side, this one, that is the medial rectus. Okay, and oblique, you see the inferior oblique, and this is the tendon of superior oblique. So oblique muscles move the rotate the eye. If superior contracts, your eyeball will move up when you look upwards. If inferior rectus contracts, eyeball will move down. If lateral contracts, it will move the eyeball lateral. If medial contracts, it will move medially. Now, these extrinsic eye muscles are also called extraocular muscles. Okay, or extrinsic eye muscles. Now, these muscles are controlled by three cranial nerves. What are those 3 cranial nerves? The main one is oculomotor, cranial nerve number 3. Its name is telling you oculomotor. Oculo means eye, and motor is movement. So oculomotor is the movement, that the cranial nerve that helps to move the eye. the main one, and oculomotor controls 4 of those 6 extrinsic eye muscles. Okay, medial rectus, superior rectus, inferior rectus, and inferior oblique. So 3 rectus and 1 oblique. These are controlled by the oculomotor. Lateral rectus, however, is controlled by a different cranial nerve, which is cranial nerve number 6 called abducens. And superior oblique is controlled by another cranial nerve, that is the cranial nerve number 4 called trochlear nerve. Okay, so those are 3 cranial nerves: oculomotor number 3, trochlear number 4, and abducens number 6. They control those muscles. That means they control the movement of the eye. There is a clinical condition that is called strabismus. uh, in this clinical condition, the person's eyeballs or eyes are not aligned correctly. Okay, so the alignment of the right and left eyes are off, somewhere else, a little bit off from that object. Structure of the eyeball: inside the eye you have 2 cavities. The anterior cavity is in front of the lens of the eye, which is smaller, that is called the anterior chamber. And there is a, so this is the eyeball, and let me draw the lens, ciliary muscle, and this is the lens. Okay, and ciliary muscle. So this is called the lens, and this is the cornea here. Cornea is here. Okay, and now in front of the lens, this small chamber is the anterior chamber. Okay, anterior chamber, and behind the lens, this big chamber is the posterior chamber. And 2 chambers contain 2 different types of fluid. In anterior chamber A, remember anterior, the fluid is like water, water-like fluid. That's why it is called aqueous humor. Humor means fluid. In the posterior chamber, you have jelly-like fluid. Okay, and that is called vitreous humor. Now, if you see, this is the lens, and lens are actually held there by the muscles called the ciliary muscle. Okay, and we'll see that. And in front of the lens you have the iris here. In the center of the iris there is an aperture, you know that. So this is the pupil and this is the iris. Okay, so you have pupil here. We'll see that in next picture. Just know that there are 2 chambers, anterior and posterior. Now in the wall of the eye you have 3 layers. So again, this is the eyeball, and outer layer is called the fibrous, which is very tough. You know, more fiber means tough. more tough. So this is fibrous outer layer to protect mainly. Then you have a middle layer, and that middle layer is called the vascular layer. So this is the middle vascular layer. Okay, and why it is called vascular layer? Because in this layer you have a lot of blood vessels. That's why middle vascular. So vascular layer. Then you have another layer, that is the innermost layer, which is the sensory, also called sensory neural layer, but this layer is only present in the back part, that means the posterior innermost layer. It is not in the front, it's like here, only in the back part, and that is the most important layer. Why? Because retina is in that layer. Okay, so that is the sensory, also called neural layer. Okay, okay, now let's go back to the outer fibrous layer. Outer fibrous layer forms two structures. The most anterior 1/6 part, if I just take 1/6 part uh, uh, of the most anterior of the fibrous layer, that is transparent, and that is called the cornea. So cornea is a part of the fibrous layer, but which part? Most anterior 1/6 part. Highly transparent. Highly transparent. So light can easily pass through it without any obstacle. Okay, then rest posterior 5th, 6th part. That means most part here is white, not transparent, and that is called sclera. Okay, 5th, 6th part. So these are two structures belong to outer fibrous layer. One is cornea here, and then most part is the sclera. Okay, so these are two structures of the fibrous layer. Then we'll see the vascular layer. Vascular layer is the middle layer, and it mostly contains the blood vessels. And where you have the blood vessels in most part of it, like here, in most part of it you have blood vessels, and that is called choroid. So choroid is the most part of the vascular layer where you have the blood vessels. However, this layer, when it goes to the front part of the eyeball, what happens, you see, it forms a muscle, and that muscle is called the ciliary muscle. So like this. So this is go to the front and forms the ciliary body or ciliary muscle, same thing. So ciliary body or muscle, same thing. Okay, here smooth muscle, and from the ciliary body, suspensory ligament arise, and those ligaments hold the lens here. So you see, this is the lens of your eye. Lens of your eye. So ciliary body uh, holds the lens in the right place inside the eyeball, and from the ciliary body actually some ligaments arise and get attached to the lens, and those are called the suspensory ligaments. Suspensory ligaments, okay, also called zonular ligaments. Anyway, now so that is the middle layer. So middle layer has the choroid, which is the most part of it, filled with blood vessels. You have ciliary body, which is a smooth muscle, and you have suspensory ligament that arise from the ciliary body or ciliary muscle, and then another structure important also formed by the middle layer. So middle layer also goes like this. You see here, goes like this and forms the iris. So this is the iris here, and you know in the center of the iris there is a hole that is called the pupil. So this is the pupil here, and this is the iris. And this hole is the pupil. Okay, so when the iris the muscle, iris is actually muscle, it has two different types of fibers. We'll talk about that. So the iris can control the size of the pupil. When too much light falls on your eye, the pupil gets smaller to limit the entrance of the light. When you walk in the darkness or in dim light, then pupil gets bigger. So that is the middle layer. So you see in the middle layer you have a number of structures, right? So these are the structures in the middle layer. Then the innermost layer, I told you, only present in the posterior part of the eyeball, and this is the most important layer or structure of the eye, because this sensory or neural layer is actually the retina of your eye, where you have the photoreceptors, right? So retina has rods and cones. So these are the photoreceptors, and the retina is attached to the inner surface of the eyeball. And this chamber, we talked about that already before in last slide. This chamber is filled with jelly-like fluid that is called the vitreous humor, and that actually gives the shape of the eyeball and maintains the shape of the eyeball. That pressure of vitreous humor keeps the eyeball round. Number one. Number two, this is also very important because of that pressure of vitreous humor, the retina stays attached to the wall. So it keeps the retina attached to the wall. Sometimes if that pressure decreases, then there is a chance that retina will get off from the wall, comes off from the wall, and that is called, you probably have heard that clinical condition. We often hear that retinal displacement or detachment. Your eye, inside the eye, you see three layers. Let's quickly go over. This is the outer fibrous layer, and this is the sclera. That part is opaque, no light can pass through it. Only anterior 1/6 part, this is the cornea, which is highly transparent, so light can easily pass. Okay. Then the middle layer, this is choroid. You see the blood vessels, right, filled with blood vessels. So this part is the choroid in the middle layer. Then ciliary body here, and you see the suspensory ligaments arise from the ciliary body and get attached to the lens. So this is the lens here, ciliary body here, suspensory ligaments or ciliary zonule. Okay, and then you see the iris goes to the front, and the aperture or opening in the center of the iris is the pupil. So iris can control the size of the pupil. Okay, now this is the innermost, the most important layer, sensory or neural layer, which is the retina of the eye. Okay, now when the light passes through the cornea, you see when the light enters, uh, into the eye, if these are light rays, you see here light rays, uh, when the light passes through the cornea, slight refraction or bending of the light occurs, very little. Okay, and then the light will pass through the actual lens. So this is the lens, and the function of the lens is focusing the light exactly on the retina. So focusing the light where? Exactly on the retina of the eye. So converge the light. Okay, bend inwards. That is the convergence of the light to do what? Focus exactly on the retina. If the light is not focused by the lens exactly on the retina here or here, then you won't see the things clearly. If your lens focuses or meet the light here, points the light here, you won't see clearly. Or if it goes like, all the way to the back, you won't see it. So to see clearly, your light must be focused exactly on the retina. And you see from the retina, the optic nerve arise. So this is the optic nerve, cranial nerve number 2. Okay, so that is the function of the lens, focusing the light on the retina. On the retina to see the things clearly. Now, as I mentioned that the bending of light, which is called the refraction, occurs slightly in the cornea. However, mostly when the light passes through the lens. Now, the shape of the lens in human eye, it is what? Convex, called biconvex, round. So, lens is in human eye biconvex, like this. And the biconvex lens, as I told you, that refracts the light. So when the light passes through it, what happens? Convergence occurs. So this is the retina here, okay, in the back of the eye. So retina. So the lens focuses the light exactly on the retina here. So that is the refraction. Now, the shape of this retina changes all the time. And when you look at something, you know that the object, for example, this is the object here. Okay. Now, when you bring the object close to your eye, what happens? The size increases. You know that, right? So it gets bigger. The size gets bigger. Same object. So this is far, this is near. So when the object, you move the object closer or near, what happens? This lens gets more round, more round. And when you take away, it gets less round. That means more flat. Okay. So this lens, biconvex lens, always changes its shape. Okay. So near, if the object comes near, it gets more round or convex. Okay. Far, the object gets smaller, less round. Why? When the object is big, you see the light enters into the eye like this, and when it passes through the lens, it needs to be more refracted to put on the retina. When the light comes from the far object, like a small object, small picture, then less refraction or bending is needed to point on the retina. Okay, so more round lens can converge the light more. That means bends the light rays more. Less round, converge less. The goal is putting the light exactly on the retina. That's why the shape of the lens changes all the time. We talked about the ciliary body, which is formed by the middle layer, right? And from there, suspensory ligament or ciliary zonule arise, and they hold the lens in position. We talked about the iris. Iris has a hole inside, that is the pupil, and iris regulates the size of the pupil to control the light, how much light will enter. In bright light condition and close vision, the pupil constricts, gets smaller. In dim light, dilate. Here you see this is the iris, and I told you that in the iris you have two types of smooth muscle fibers. In the middle part you have the round or circular fibers like this. So circular fibers in this part, and in the outer part you have radial fibers or longitudinal fibers like this, you see. So all these fibers are like this. Okay, so two types of fibers. The constriction of the round fibers, those are in the middle portion of the iris, will do what? Will make the pupil smaller, because this is round muscle, so when it will constrict, the fibers will constrict, the pupil will get smaller. And when these muscles, radial or longitudinal muscles, contract, they pull the circular muscle from all different directions outwards, so the pupil gets bigger. Okay, so that is the uh, dilatation of the pupil. So that's why you have two types of muscle fibers. Circular muscle fibers are more useful to constrict the pupil, and the outer radial or radial or longitudinal fibers are more useful to dilate the pupil. Here you see constricted pupil here, so the size is small, and here you see heavily dilated pupil, so the size is very big. And in different conditions, the size of the pupil changes, as you know already, that if I put flashlight, then pupil will constrict. That is called pupillary reflex of the pupil. We often test the person's consciousness by putting flashlight on the eye to see if the pupil constricts or reflex is there. That indicates the motor function is working, is functional. And in dim light condition, the pupil dilate, because in dim light the light is less outside, so pupil will get bigger to take as much light it can take inside to give you the sight to see the thing. Another is parasympathetic. Autonomic nervous system has sympathetic and parasympathetic. Parasympathetic stimulation will constrict the pupil. Probably you know that parasympathetic stimulation, when the person is in comfort condition or comfortable condition, parasympathetic stimulation occurs more and the pupil gets smaller. When the person is in fight or flight situation, and excited or scared, then pupil gets dilated because sympathetic stimulation occurs in that condition. Now, the retina, the most important structure of the eye, and if you see the structure of the retina, it has two layers: pigmented layer and neural layer. Pigmented layer is the outer layer, and this layer absorbs light, and so when the light falls on the retina, it will not be reflected. If light falls on the retina and reflected, then the photoreceptors will not be activated. So to activate the photoreceptors, you need the light to be absorbed in the retina. If it just falls and immediately leaves, then the photoreceptors will not be activated. So uh, absorbing the light is the function of the pigments of the pigmented layer. And also this layer has vitamin A stored. Now the inner layer is the neural layer, and this layer is more important. Why? Because the photoreceptor rods and cones are located here. Okay, so these are called photoreceptors. However, in the neural layer you have few other cells: bipolar cells, ganglion cells, amacrine cells, horizontal cells. All these cells are important to uh, converse the light. So here you see this is the retina, and only a small part is the pigmented layer here, very thin. You see here, this thin layer here is the pigmented layer. The pigments are here. So that is the pigmented layer. This is the middle layer, choroid, so which is vascular layer, you know that. So that is not a part of retina. This is the pigmented layer, and then neural layer is much thicker here, and here you have the photoreceptors. These are photoreceptors. You see yellow are the rods, and green, blue, these are the rods. So you see it by two different colors, the rods and cones. And now you see this is the part of the retina where there is no photoreceptors here, right? It's actually a hole in the retina. Nothing is here. So this area is called optic disc or blind spot. So since no photoreceptors here, your retina cannot process any light here. So you should actually see a hole when you see at something, because the picture, if falls here, there is a gap here, right? No photoreceptors. However, since you have two eyes, that compensates that loss. There's a filling effect, so you don't see that, and your eyes are always shaking. That's also helps to fill up that gap. Anyway, so that is the optic disc where there is no retina or photoreceptors. That is why it is also called blind spot. Then uh, from that area, the optic nerve emerges or gets out. So optic nerve is attached to this area. So this is the optic nerve, cranial nerve number 2, and also the blood vessels pass through that area. So blood vessels enter into the eyeball through the optic disc area, as well as the optic nerve is attached, and optic nerve takes the electrical signal out from the retina to the brain. Okay, now you remember I said that in the retina neural layer you have rods and cones as well as other neurons. So you see other cells or neurons are here. We will see what are those cells. Now here you see this is the pigmented layer. Here you can see pigmented layer here. See the pigments, absorb the light. So the light will stay there, will not be reflected, and just under that you have the cones and rods. So the rods are rod shaped and cones are cone shaped. That's why they are called rods and cones, and they are activated by the light. Okay, and the action, the activation of rods and cones will transmit the electrical signal. Okay, and from the rods and cones, you see these cells receive the signal here. These are called the bipolar cells. And then you see from the bipolar cells some horizontal cells. These are called horizontal because they are like getting the signal horizontally like this from the rods and cones. So you have horizontal cells, bipolar cells, and another type of cells. These are called amacrine cells. So they basically uh, receive the signal and take the signal from the rods and cones to these cells or neurons. These are very important. These are called the ganglion cells. So ganglion cells finally receive the signal. So from the rods and cones, the signal finally uh, reaches here in the ganglion cells. Now ganglion cells are neuron type cells. So you see these are actually dendrites and this is axon. So now you see all the axons of these ganglion cells, they actually join together, bundle together and get out as the optic nerve. So these axons, all these axons are bundled together. Axons of what? The ganglion cells. And that is the optic nerve. Optic nerve. So let's go back to previous slide here. So this is the optic nerve here, which is actually the bundle of the axons of ganglion cells, remember. Okay, and the action potential, you remember repolarization, action potential is generated in the ganglion cells, and that shoots the signal through the axons towards the brain. So the optic nerve will take the signal towards the brain or the central nervous system. Okay, so that is how the signal is processed in the retina and gets out. Now if I see the retina, this is actually the real picture of a retina. You can take the picture through the pupil by dilating the pupil. You know, sometimes the doctors do that. When you go to the eye doctor's chamber, they will put a drop to dilate your pupil a lot, and they can take picture from the back of the uh, eyeball, the picture of the retina. Now you remember the area where there is no retina, no photoreceptors, the blood vessels enter and optic nerve is also attached. That is the optic disc, also called the blind spot. So you see no photoreceptors are here, no retina. Now if you see the center of the retina, in this part you have two structures in the center of the retina. One is a tiny spot in the center that is called the fovea centralis. Fovea centralis. Very, a small area in the center of the retina, and around the fovea centralis you have the area that is called the macula lutea. Okay, so now if I see the fovea, this is the retina. Okay, this is the retina, and let me draw the fovea centralis and macula lutea here. Okay, so this is the retina, outer part of the retina. This is the macula lutea in the center part, and the most central small area is called the fovea centralis. Now, in the center part of the retina, you have the cones in highest density. So the cones are located in the center, and rods are located in the outer part. So rods in the outer part and cones in the central part. Now there are three types of cones. So in the center part you have the cones, and there are three types of cones. The red cones, those are not actually, the color of the cones is not red. Those are called red cones because they absorb red color light or red color. So that is one type of cones. There is another type of cones, they absorb, or they are stimulated by the green. So red, green, and another type are blue cones. Okay, so blue cones, RGB, red, green, blue. So those are three types of cones. Cones are red, green, blue light sensitive cones. Okay, and they are located in the center part. However, in the fovea centralis, the cones are present in highest density, very densely present, those three types of cones. Okay, heavily packed. Now remember, to see something very clearly and small, you need to look straight, and the picture must fall in the fovea centralis. Okay, the picture of that object must fall on the fovea centralis, and when it will fall on the fovea centralis, when you look at something straight. So the light will go straight and will go to the center here. Okay. Therefore, to see something small and fine detail, which is called the visual acuity, you need to look straight to put the picture on the fovea. Okay, anyway, so those are three types of cones. Now if someone has red cone deficiency, it is usually a genetic disorder due to lack of that gene. Someone might not have enough red sensitive cones, or even it could be completely absent, and that is called red color blindness. If someone has green cones absent, green color blindness. Some people have both red green blindness. Okay, so the color blindness or color deficiency due to lack of color sensitive cells, those are the cones. And rods are located in the outer part, the peripheral part of the retina. So these are the rods. Okay, so these are the rods. Okay, so that's how the rods and cones are located in the retina. Now I'll mention the important functions of the rods and cones. First, the rods in the outer part are responsible for dim light vision. That means to see the things in lack of light or less light condition. Okay, and cones are responsible for a few things: bright light, vision. That means to see in bright light, your cones are more useful, not the rods. That means to excite the cones, you need bright light, more light, so less sensitive to light. Then color vision. I already mentioned that to see the things in color, you need the cones. And we see more than 3 different colors, right? We see other colors too. Why? Because when 2 or more of these 3 colors sensitive cones are excited, then you see the combination of these 2 colors, right? If you excite the green and red cones, then you will see orange or yellow. And then another is the visual acuity. What is visual acuity? To see the things very clearly, small and fine detail of something. Acuity. So these are 3 important functions of the cones. Okay. We already talked about the lens. Lens is biconvex, highly transparent protein inside it. That protein is highly transparent and flexible. I mentioned before, that changes the shape, and avascular, no blood vessel, which is important. If there are blood vessels, then the light will be obstructed or blocked. So that's why the lens doesn't have any blood vessels inside it. It is avascular. That's why you can do surgery easily without bleeding, enough bleeding. Allow precise focusing the light on exactly on the retina to see the things clearly. uh, Now the lens could become denser and cloudy and less elastic with the age. So in older people, the the transparency of the lens may decline. The lens could become cloudy and less elastic. That means the flexibility of the lens will be less, so it will not be able to change its shape quickly. So that will cause the problem in vision, right? And that condition, cloudiness of the lens, is called cataract. So this protein gets dense and cloudy. So age is one factor of cataract, but there could be other factors that can cause cataracts: diabetes mellitus, heavy smoking, frequent exposure to UV radiation. So here you see cataract, okay, cloudiness. Now the refraction problem or problems of refraction. Uh, if your lens, you already know that lens focuses the light exactly on the retina. I mentioned that, right? This is the retina. So the light, when it passes through the lens converges the light to focus exactly on the retina. Now, if your lens cannot do that, if the light is focused in front of the retina, not exactly here on the retina, or behind the retina here. In both cases, the person will see blurry, will not see clearly. To see the things clearly, the light must be focused exactly on the retina, right? So that is the refraction. Those are the refraction disorders or problems. One is called myopia or nearsightedness. The person can see the near things clearly but cannot see the far things. Okay. Uh, what causes myopia or nearsightedness? If the focal point is in front of the retina here. So this will cause myopia. Hyperopia, farsightedness. The person can see the far things clearly but has problem in seeing the near things like reading or seeing the near things. That will occur if this happens. The light is focused far behind the retina. So that is hyperopia. Or farsightedness. Now what you have to do, since the lens cannot focus the light on the retina to see the things clearly, what you have to do, you have to, if this occurs, myopia, you have to push that light backwards to put on the retina. If this happens, then you have to move the light forwards to put on the retina. The goal is adding extra lens here, which is the glasses, eyeglasses. Okay, eyeglasses. We put eyeglasses to either push the light focus on the retina backwards on the retina, or move the focus from the back to the front. The goal is putting the light exactly on the retina. So that's why we use two types of glasses. In myopia, we use, since it is here, so we use concave to push it back, and to bring it forward. In hyperopia, we use convex lens. Okay, so these are two lenses we use in two different disorders or problems. So this one convex and this one concave. Astigmatism is another condition of refractive disorder in which the surface of the lens gets irregular. So this is the lens and the surface of the lens gets irregular, or the cornea, surface of the cornea gets irregular. So unequal curvature in different part of cornea or lens. Here you see this is normal eye, which is called emmetropic eye, normal. The lens focusing the light exactly on the retina. Here you see the light is being focused in front, so you have to move it back. So you use concave lens. This myopic condition occurs very often. You will see in young kids when they suddenly grow, their body suddenly grows. What happens, the eyeball starts to grow faster than the orbital fossa, the bone around the eyeball. So a pressure is created on the eyeball, and eyeball gets elongated this way, longer. This is the actual shape of the eyeball here. The retina is actually here. Since due to pressure from outside, eyeball gets longer or elongated, retina moves backwards further, right? So your lens doesn't know that, it will try to focus here. So that causes that refractive problem. When you put the glasses or extra lenses, it will push back here. This is opposite condition. Here you see the focus is behind the retina, and you have to use convex lens to bring it forward here. So that is hyperopia. The last thing is the visual pathway. So you already know that optic nerve is formed by the axons of ganglion cells, right? Bundle of axons of ganglion cells. Two optic nerves here, you see, from two eyeballs join here at optic chiasma, where some fibers, those axons, cross to the opposite side, which is very important. So from your both eyes, your both halves of the brain get signal. You see some fibers cross, some fibers stay in the same side, right? Same thing here. Some fibers cross, some fibers stay in the same side, and they bundle together, the crossed and same sides fibers, to form optic tracts. So optic nerves, these are optic nerves, optic chiasma, where the crossover takes place, and then they bundle again to form optic nerve. And from optic nerve, the signal is relayed, taken to the thalamus. Okay, and from the lateral geniculate nucleus of the thalamus. You know, thalamus is the major sensory relay station, so sensory signals are relayed here. And then from here to the primary visual cortex by the optic radiation. These fibers are called optic radiation. Okay, so let me go over again quickly. Retina, optic nerve, then optic chiasma is the crossover, then optic tract, thalamus, then via optic radiation to the primary visual cortex in the back of the occipital lobe. Okay, so that is the visual pathway. Let's stop here. So for visual pathway, just know those structures from the retina to the primary visual cortex.